All right, so in this video, I'm going to be talking about proteins and really just the basics on what is a protein, kind of how they fold, um, how they're synthesized is going to be in chapter 17. So this is just going to be talking about first and foremost, what is a protein? And that's really kind of a massively loaded question when you think about biology in its entirety, because proteins make up pretty much everything that is a cell for the most part that you would be able to see in terms of how it receives a signal, what gives the cell its identity. So proteins are going to be kind of a massively important topic throughout this, really your AP test and just this class in general. Um, so it's important to just talk and review what are proteins made up of, and this is just showing you an actual monomer of what is a protein, and this whole entire thing is going to be called your amino acids. So proteins are made up of amino acids. So this is one amino acid. On the left side, you have your amino group. On your right side, you have your carboxyl group. And so this just shows you have your amino, your alpha carbon, and then your carboxyl group. And then your R group is going to be your variant. So all amino acids, regardless of what kind it is going to share this amino carbon carboxyl. The R group is going to be what's able to be changed. So when you think of what is an amino acid, it's this, and the R group changes. So when you think about the amino acids, this is what we're looking at. So there's only 20, but the amount of proteins that we can make is pretty much endless. So it's just about the order of these things. So just again, all of these share this component, your amino, your carbon, your carboxyl, your amino, your carbon, your carboxyl. So if you look at the R group, that's what's going to give each amino acid its identity. So in terms of what we're looking at, these ones in green are all going to be non-polar. So these are all, and when you look at the R groups, they all have some similarities in the sense that they're primarily made up of hydrocarbons, which is just carbon and hydrogen. Not something you have to know for your test, but you should be able to identify, okay, that these R groups is what gives this amino acid its identity. So when you look at the names, for example, if we look at glycine, the R group is just a hydrogen. The name of this one amino acid is glycine. It's abbreviated by the letter G. There's also another abbreviation form, which is GLY. So biochemists get lazy, so oftentimes they don't, when they're writing out the actual order of the amino acids that make up the protein, they're either going to just use GLY or even get even lazier and just have a G. So not all of it is going to make the most sense. So like aspartic acid, it makes sense that ASP is going to be the abbreviated form, but capital D is going to be the abbreviated form. So these are just the 20 amino acids. There's actually 22, but for the most biologically important enzymes and things that we're going to be discussing, it's all made up of these 20. And it's just how they're arranged is what gives this protein its identity. So that's kind of interesting. So when you think about it, what do proteins do? And that's where it gets kind of crazy, especially now it's towards the end of the year. We've talked about enzymes in terms of transport proteins, hormones, receptor proteins, and cell signaling, and kind of their function. It's pretty much all of everything that we've talked about throughout this year is all based off of these proteins. So in terms of that, here's just a short little recap. Oh, let's see what this one is. So it looks like this one is talking about that as a transcription factor. So basically this purple protein is going to help the DNA be expressed. So this would be a regulatory protein that's actually going to help make another protein. So this is just showing you all of these things in purple are different classes of proteins. I don't think I have all of them animated, but I think this next one is going to be a cell signaling one. Yeah. So there's the signal, binds to your receptor protein. So this is all just kind of an overview that each one of these is a channel protein. Proteins are endless in terms of their capabilities in the cell. So one classic example that I really need to kind of point out, especially with this chapter, is oftentimes when you think of proteins, a lot of people like to just draw this cartoonish drawing, which is really, really important to get the point across that this site, especially when we talk about enzymes, this is called the active site. And that's where your substrates bind to. So this is just one example, one class of a protein that we've discussed. So just showing you that enzymes are just one category of a protein, but the shape of each enzyme, is specific to its function. So this just kind of shows you other variants of proteins, like we have enzymes, we have antibodies, which we didn't get to the immune system, storage proteins, we have channel proteins when you think about like osmosis, not only letting water in, but letting ions back and forth. This is talking about cell signaling, specific hormones, neurotransmitters, muscle contraction, as well as actual structural proteins, things like collagen and keratin. But when you think about a protein, like I said, a lot of books like to just show this as the protein, but really when you look at them, this is what they look like. So this is showing you the nice folded structure of a protein. And let's just kind of get into the vocab here is what is a protein? And that's a functioning molecule. And it consists of one or more polypeptides. So a lot of people struggle with the concept of what's a polypeptide, and poly is essentially meaning many, many peptides. So right here, this is showing you that, okay, here's these abbreviated forms of those amino acids like GLU. If you wanted to go back to this, GLU would be glutamic acid or glutamate, which is down here. So basically what they're trying to show here, rather than draw out that whole drawing, they're just saying GLU is glutamate, right? And then it starts to fold. So let's get back into that. This is one polypeptide they're showing in blue, but there's another polypeptide that it's bonded to. And this is the functioning, this whole thing would be the functioning protein. So just to kind of reiterate, here's the whole 20 amino acids, not something you have to know. Do know the backbone for sure. But this is just going to basically get into how proteins fold. That's going to be the main focus for this video. So in terms of this, you want to understand that each one of these 3-letter abbreviations is representing one amino acid. And they're going to be held together by peptide bonds. So carbohydrates are held together by glycosidic bonds. Peptide bonds, I would say there's a peptide bond here, 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 here. And each end of the polypeptide has one of those amino ends and a carboxyl end. So when you think of this, there's no limit on how long a polypeptide can be. It's usually going to be determined by how long the DNA is. So the DNA is going to be read to make this amino acid, this whole functioning polypeptide. So let's look at how these things actually fold, or not fold, I'm sorry, how they make a bond. So right here, I have 3 amino acids. I have this amino acid with this R group. I have another amino acid here with this R group. And then what I'm trying to add in is another amino acid. So when you look at this picture right here, I'm trying to add this amino acid to this polypeptide. And so you're seeing this common theme a bunch where anytime you're making a bond, you lose water. So that's your dehydration reaction, you're losing water. And right here is your new peptide bond. So if I were to ask you how many peptide bonds do you actually see in this protein or this polypeptide? Well, I have a peptide bond here and I have another peptide bond here. So if it's 3 amino acids long, you're going to have 2 peptide bonds. If it's 4 amino acids long, it's going to be 3 peptide bonds, so on and so forth. And this is a good overview to show you that you always have your amino and your carboxyl end. And that's another big thing that I talked a lot about in class. You can only form a peptide bond between a carboxyl group and your amino group. Oftentimes your multiple choice questions or even an FRQ, for example, would have it to make sure that you guys understand you can only form a peptide bond between those 2 functional groups. So the orientation of how these things line up when they make these bonds matter, meaning the carboxyl group has to always be next to your amino group, and that makes your peptide bond there. So this is just showing you how this bond is formed. Again, a common theme, it's a dehydration reaction. You're losing water, there's your peptide bond. And I have 3 amino acids linked together here. So in terms of the proteins, like this is really what they look like when they're all folded and convoluted. So what I just showed you, that making that bond was the primary structure. The rest of this video is just going to be talking about your secondary, tertiary, and your quaternary folding. So secondary and tertiary is where it actually folds and based off of the R groups. And then when we get to the quaternary, that's when we look at multiple polypeptides. So let's kind of get into this. This is really simple and just showing you that if it just gives you the order of the amino acids, this isn't really folded like this. This is just showing you to like make room on the slide. But it's just the order. So what's the order of these amino acids? So like this one, I have glycine, proline, threonine, glycine. So this is just showing you the actual order of amino acids. So don't forget how this was made. Remember, we talked about DNA, when you actually read the DNA to make the RNA to make this protein. Remember, every 3 nucleotides is going to code for 1 amino acid. So nice to kind of revisit that at this point. So you got your amino end, your carboxyl end. This is the primary structure. And it only exists like this just for a short deal, and then it starts to fold right away. So here's a short little animation explaining the primary structure. So if you take a closer look at this, it's going to show you a more up close representation of what it looks like at the molecular level. Speed this thing up. Yeah, so it just shows you when you zoom in, there's your carboxyl group, there's your amino, there would be your peptide bond, there would be your R group. And so what I like about this little animation is it's just reminding you that it's the R group that can change. So when you look at this, you don't have an R. It's just showing you that that's like it's like a variable in math, right? It changes. So it's specific to what the gene was on what amino acid is put there. So just showing you that that R group can change, and that's going to give the amino acid its characteristics, whether that's polar, non-polar, whatever have you. So once you get that primary structure, it's then going to start to fold. And the biggest thing that you guys need to know, the first time it folds, it's just based off of the backbone and the backbone of the carboxyl group and the hydrogen of an amino group. So basically, it's just with hydrogen bonds. So remember, hydrogen bonds can only really exist between hydrogen and oxygen, hydrogen and nitrogen. Primarily here, you're just going to see it between the hydrogen and oxygen. So the POGL, I'm pretty sure I have some pictures here. They do a really good job explaining this. But again, this is a nice representation that it can fold like this helical structure called an alpha helix, or it can kind of go back and forth, and that's when it forms a beta pleated sheet. So this is showing you one polypeptide, one protein where it's showing you that this one has an alpha helix and this one has the beta pleated sheet, and it's a single polypeptide. So looking at this one, this shows you how it's going to fold. So it shows you how that folds based off of the secondary structure. And so I think it's going to show you the alpha helix first. Now there's other characteristics we'll get into in a second on what also contributes to the folding of the protein. But remember, the R groups don't really play a role in this part. It's just within the backbone. So it doesn't matter what your R groups are, you're going to have some secondary folding regardless. So yeah, just showing you that it's between the hydrogen on an amino group and the oxygen of one of those carboxyl groups in the backbone. So if I didn't talk about the backbone, let's remind you what that actually is. So because all of these amino acids here share this characteristic, the amino, the carbon, the carboxyl, the amino, carbon, carboxyl, that's what we refer to as the backbone. The R groups aren't considered part of the backbone. So these are your R groups. What's in purple is going to be referred to as the backbone because they're all the same. So that's the first time it folds. But when you get into the tertiary structure, that's where things get a little interesting. It's when those R groups start to interact. So it's already folded with those hydrogen bonds in the backbone, but then it's actually going to fold again according to the R groups. So in terms of this is really the second time it folds, and it's based off of a couple of similar characteristics that we've talked about earlier. This is showing you that you have a hydrogen bond here. You have two non-polar interactions here. You got your disulfide bridge. Everybody loves that one. And then you got some ionic interactions here. So it's just a way that these R groups can fold. So if you look at this one, this is going to show you that it's already folded and it folds again. And so when you look through this, zooming in, it kind of basically characterizes how it folds. So if you look at the blue represents the hydrophilic or polar interactions. So these two both have a charge, so they really want to be next to each other. This is showing you the hydrophobic or non-polar interaction. So you think about it, non-polar wants to be next to non-polar R groups. And I had shown you guys this a bunch in class, so I pulled this up here. If I were to essentially reset this, let's generate a random protein. This is just showing you like the primary structure right here, but we are going to let this play out, and I want to look at all of the charge and the hydrophobicity. So the goldish color represents non-polar interactions. The green are going to represent polar R groups, and then the purple I'm pretty sure is positive, and the glutamine is going to, or the pinkish is going to be negative. So there's going to want to be some type of attraction here. So if you let this play, notice what you're seeing here. And it's really, really, really important that you can see the difference. So this is showing you that the solvent or the solution that we're in is all in water. So this whole protein is in water. All of these gold or non-polar amino acids are all trying to kind of get next to one another. And that makes sense because non-polar things don't want to be next to a polar environment. So that's why all these non-polar amino acids are kind of tucked in. You're going to see all those polar ones exposed on the exterior. And again, it's never going to be perfect, right? Because things are constantly moving. But these polar amino acids are okay being on the exterior. And that has to deal with the polarity. So these guys are okay. There is some charge. All those non-polar ones are always wanting to kind of orient themselves on the interior. So let me just regenerate another random one, and I'll do the same thing. And just kind of double check, okay, all the non-polar ones want to be in the center or the core. They want to escape that polar environment that they're in. And again, it's limited to what the primary structure is. So these ones are kind of going crazy just because you can't change the primary order, but they're still doing their best to kind of be as satisfied as possible. And I'm going to show you one other one. I'm going to generate a random one, but rather than put it in water, I'm putting this protein in a solution that is non-polar, so it's an oil. So if I let this protein fold in this environment, let's kind of take a look and see what happens. Notice now that all of my non-polar amino acids have folded, and those are the ones that are okay on the exterior. All of my polar R groups are going to be on the interior. So pretty interesting and simple representation of how this protein folds. And so now when you go back, hopefully this makes a little more sense. If I played this out again, where if I let it fold for the tertiary interaction, it's folding, and it's folded based off of what those R groups are. Now it's not showing all of them here. It's just showing you all the different types of interactions. And once you get that folding, it's going to then find another, if it is required, another polypeptide to make up the complete protein. So this is showing you each color represents a polypeptide. So I have 1, 2, 3. I got the green polypeptide, the blue polypeptide, and then the pink or peachish color. So this one would actually have three polypeptides linked together. So in terms of that, most proteins have a quaternary structure. So when you see this, it's just going to be representing that this polypeptide combined with this polypeptide is going to make the actual functioning protein. So things like, I think it talks about hemoglobin. Hemoglobin has 4 polypeptides in its quaternary structure. So basically that just means with this polypeptide by itself, it's not going to do anything. But if I have all of these different polypeptides linked together, that gives it its functioning characteristics of hemoglobin. So, and here's another representation of it. So I got all this folding. I got a couple alpha subunits, a couple beta subunits, just showing you that this gives this protein its complete identity and complete functioning characteristics. Now, here's just another short snapshot of how this folds. If this is showing you all different characteristics of these amino acids, it's showing you that, okay, I can make a disulfide bridge here. I have some ionic interactions here. And this is just showing you different R group interactions. So anytime your R groups are interacting, that's going to be representative of your tertiary folding. So it really is like a sequence. So what I have here is just a couple of snapshots or screenshots from your POGL that we did a long time ago. Again, here is where you're going to do your dehydration reaction. And this is where your new peptide bond is going to be formed. So if I were to ask you point blank how many amino acids make up this polypeptide, you would see 2. So kind of interesting and good representation. It's the dehydration. I formed water as my byproduct here. And again, this is where I really think that this POGL does a fantastic job on their animation. So this is showing you what's the primary structure. It's just the order. But this kind of helps get the point across. What is the backbone and how does it fold? So this right here, everything that I'm circling, not the R group, this is the backbone. So it's showing you that there is going to be some interaction on the backbone. Here's a hydrogen bond. So let me actually go ahead and erase that. Yeah, there we go. So if you look at this, one common misconception is, well, can I form a hydrogen bond here? And the answer to this is no, because if you look at it, it's not between this carboxyl is okay, but you're forming a hydrogen bond here with this alpha, this carbon on the R group. That is not acceptable. That's not stable. So really, if I were to say draw in, or if your AP test for that matter said draw in an appropriate hydrogen bond, it would have to be between this hydrogen and that oxygen, because that is going to be the amino group here, and this is going to be the carboxyl. And notice that they don't have to be right next to each other. Oftentimes they're pretty far down. So each line or each dot represents the hydrogen bond. So that's what I'm showing you here. And just to kind of show you again, here would be another appropriate hydrogen bond, filling that in just like that. That's a good hydrogen bond because you got your amino and then there's a carboxyl on that one, okay? So this is just showing you it can either form that beta pleated sheet or the alpha helix. And this is showing you the tertiary R interactions. Again, nonpolar want to be next to nonpolar. It's an ionic, hydrogen bonds, and then your disulfide bridges. And if I didn't talk about it, I'll mention it now. Here's 3 different polypeptides. The interactions that they have is going to be the same as your tertiary. So this just shows you I can have a disulfide bridge here, an ionic interaction here, nonpolar interaction here. So just representing what holds them together, it's the same interactions as your tertiary structure. So this last little bit on proteins I want to mention is mutations, and really what those are all about. I have a whole nother video in chapter 17 about them, but this just shows you that if you change the primary structure, you're actually going to change the final folded process of that protein. So, excuse me, there is a difference between if you change that primary structure, you're actually going to change the secondary, tertiary, and really even quaternary structure of it. So this would be normal. If you change just one amino acid, if that's going to represent in a faulty beta subunit, and that's going to definitely change the shape and functioning characteristics of that protein. So that's just one way that the actual final and quaternary structure can be changed by a genetic mutation. Now, don't get that confused. Don't say that this is a denatured protein. It's not. That's a genetic mutation. When you think of things like really denaturing, where you actually change the shape of the protein was actually folded correctly. And if you changed environments, things like the temperature, the pH, that can actually break apart all those tertiary and quaternary bonds, and that can cause some major issues. So the difference is that if it's denatured, the protein was folded correctly, but due to the environment that it's in, the protein has now changed shape and it's lost its characteristics. So chaperonins are wild. It's just basically a protein that helps assist in the folding of those proteins. Other things than that, this is just showing you that here's your primary structure. It goes in, and then it basically provides a perfect microenvironment for that protein to fold. So in terms of that, the biggest thing, what is a denatured protein? Why does it no longer function if it is denatured? And the biggest thing is that if you change the shape, you're going to change the functioning characteristics of it. So that, in a nutshell, is going to be the biggest component about denaturing. So I think this just shows you the poke again. And that concludes the basics on proteins and protein folding.